So for the next hour or so, I'm going to talk about CD spectroscopy for biopharmaceuticals, as you already realise. And I'm hovering near here just so that I can coordinate the recording at the same time with the cursor. Okay. So what am I going to talk about? So it's going to be, first of all, what is CD? Have any of you used it before? You know anything about it? No? The units that CD can be expressed in, the concept of chirality, which is essential for CD, and I'll come on to explain that, and then how it can be applied to different molecules and drugs. So I'll run through CD used to look at proteins, nucleic acids, carbohydrates, polysaccharides, and then come on to drugs and how they interact with those biological molecules. I'll talk briefly about instrumentation and how you can go about doing CD, at least in the UK and elsewhere, and then give you 3 examples of the use of CD in pharmaceutics and drug discovery and drug analysis, really, and drug preparation. So it hits a lot of different things to do with biopharmaceuticals. And finally, I've just put this at the bottom of this slide just to remind me to say that there's an assessment that goes with this lecture. So I'll come on to what the assessment is about at the end, but there's going to be useful info in this lecture and also in some associated reading, which brings me neatly on to the list of reading. The first 3 or 4 references are to do with principles of CD, really, and its application to specific biological molecules. You might wonder why there's one reference in here for archaeological wood. The reason I put that in is because it describes the principles of CD in that chapter. That's why that's in there. But also Kelly et al. Do a really good, it's a bit old now, but it's a really good review on CD as well. So the principles are covered there, and then specific biological molecules. And then the final 3 are references to do with the 3 examples that I mentioned I was going to talk about, which you can consult as well. Okay, so that's the reading. Okay, so to begin at the beginning, what is CD? So what you have in CD are 2 opposing light waves, left-handed circularly polarized light and right-handed circularly polarized light. And CD is the differential absorption of one or other of those different light sources. So when you put a biological molecule, let's say protein for the sake of an example, in the path of 2 circularly polarized light beams, if it's a chiral molecule, it is likely to absorb preferentially either the left-handed or the right-handed at different wavelengths, ranges, which I'll show you in a moment. In fact, this picture at the bottom just illustrates that. So there's a 0 line here, because if you have equal amounts of these 2 light waves coming out of the source, there's no sample in the way of that light beam, the detector will say, I'm detecting equal amounts of both. And therefore, in a control like that, the spectrum at different wavelengths will just bump along the 0 line from beginning to end, because there's no differential absorption either of the left-handed, which is represented by a positive CD spectrum. It'll move, if it's a positive value, more of the left-hand light is absorbed by that molecule, or it can be shifted into the negative CD values, and that indicates that more right-handed light is absorbed by that particular molecule. There's no pattern, there's no way of really predicting what you're going to get, but that spectrum shown here is characteristic for that molecule in that sample. Okay, so it does, because it depends on chromophoric groups, just in the same way as absorbance does, absorption of light by chromophoric groups, then it obeys the same laws as absorption spectroscopy. The Beer-Lambert law applies as much to CD as it does to absorption spectroscopy. It's just that you have two waves, and you're looking at the differential absorption of one or other of those light beams. I've mentioned that you need chiral chromophores. That's the only other difference from absorption spectroscopy. So are we okay so far with that? Is that right? Okay. And this equation just reflects that, the difference in absorbance by right and left-handed light. Two components to that. So we can express CD in that way, just the change, delta absorbance. There are other ways of expressing it, which are good to be familiar with. So from the Beer-Lambert law, A equals ECL, you can express it instead of delta A, you can express it as delta E, which normally stands for molar extinction coefficient, doesn't it, for a particular molecule. In this case, it's just molar differential extinction coefficient. So you can express it as delta E as well, in which case the units change to this, per molar per centimetre in this case. And there is a third way to express CD units based on the observation about what happens when you get differential absorption of one or other light. What happens to the resolved component is that it becomes more and more elliptical. So I'll explain what I mean by that. So the green, this is the source, and it's going to run through a sample of some sort. So at the source, the green represents the right-handed circularly polarized light, and the red the left-handed, coming out of the source in equal amounts. Hit the sample, and well, before it hits the sample, you have a resolved component of those two, which is shown here as the wave, the resolved component of those two, which is flat. And then what happens as it goes through the sample is that resolved component becomes more elliptical, as shown by the blue. So you can see that as it passes through the sample, it's becoming increasingly elliptical in shape because one or other is being absorbed more, either the red or the green. Yeah.
What do you mean by the resolved part of that?
The sum of the two, if you like. So this is a flat line because it represents zero difference. So this is representing, I suppose, a waveform which is flat. There's nothing happening there. But then you see the elliptical shape from the resolved component emerging because one or other has been absorbed more. Is that all right? And so you can express CD. Oh, sorry.
How would you make like left-handed or right-handed?
I used to have a slide to show this. So what you have is two sources of polarized light which are emitted at 90-degree differences in angle. And it's the resolved component of two light waves at 90-degree differences in angle. And the result of that 90-degree difference in the two waves is to form a circle because it's satisfying two different directions of the light, if you see what I mean. I used to have a lovely video to show that, how you generate that. That's the explanation. 2 waves, 90-degree difference. When you look at how what the resolved component of that is it's a circle. It's a circle is the resolved component. Okay. So just to expand on that ellipticity, you will see those units used in the literature that you're going to do in the coursework as well. And so the blue represents the right-handed, the red, the left-handed, and the ellipticity that you see can be defined by the angle shown here, theta, which gets smaller as the ellipticity gets bigger, as you can imagine. And you can express the CD values in terms of things like mean residue ellipticity. So if it's a protein, you know what the protein is. You can work out what the mean residue weight is for that protein. In most cases, we know what proteins we're working with. And so if you do, then you can express it as the mean residue ellipticity, which depends not just on that value of mean residue weight of the protein, depends on the concentration and the path length, just as it does for the Beer-Lambert A equals ECl. So those are the units, and a variation on that, still on ellipticity, is that you can express it as molar ellipticity. And that's just taking into account moles rather than mean residue weight. Doesn't matter which one you choose. And there's a simple way to relate ellipticity to delta E or delta A, and it's simply shown by that equation with a numerical interconversion between the two, because in the end they have to reflect the same things, so they are directly related to each other. So this graph is just to show how you can, what sort of spectra you will get in the case of absorbance and CD, shown here for a sample. So if in absorbance you get a positive peak here, and in CD you get a positively shifted spectrum here, then the positive CD spectrum with light absorbed more than the left-hand light absorbed more than the right is what that is showing. If you get an absorbance spectrum again in the positive, but a negative spectrum in CD, that means that you've got chromophoric groups where most of the right-handed light is absorbed. And what would you get with an achiral chromophore is, well, of course, in CD, it will only reflect what you're seeing with chiral molecules, so you see nothing. But of course, in absorbance spectroscopy, you'll still see that spectrum because it is absorbing through its chromophoric groups. So that's the difference summarized between absorbance spectroscopy and CD spectroscopy. Is that right? So you can express it either in terms of molarity, mean residue weight, or ellipticity. Those are the 3 main ways. Now, I've mentioned the chirality thing, and dependency on a chiral molecule somewhere in the mix. And we'll come on to talking about achiral drugs in a moment. So CD depends on chirality in solution, and this reflects the asymmetry of the molecules that you're looking at. So you're all familiar with the term of enantiomers. These are, chirality is the mirror image of 2 identical molecules. So if my hand is a molecule, this one hand is an enantiomer of the other. These are identical conformationally, structurally, but they're asymmetrical, and what that means is that they cannot be superimposed with respect to each other. These are not superimposable. though they're identical, they are mirror images of each other and therefore asymmetrical. And CD can only pick up differences in light, left and right-handed circularly polarized light, because there is that asymmetry to see that difference in CD. So we're talking about optical isomers here then. So I've just explained that, haven't I? So the physical and chemical properties of a pair of enantiomers are identical, with two exceptions. The way they interact with polarized light, isomers differ in that property, and the way they interact with other chiral molecules. So this is why CD works. So one of the reasons it's so good for looking at biological molecules is that a lot of biological molecules are based on carbon, and that asymmetric valency, as it were, of the carbon atom, which is of course more of that, the back. So it's an asymmetric, that's the division of symmetry in the carbon. And so that makes that chiral very often. And all amino acids are chiral apart from glycine. So there is an exception there. So that's what makes it so ideal for looking at biological molecules. The chances are your molecules will be chiral simply because of the carbon atom. So used for protein, DNA, carbohydrate, which I'm now going to relate the specifics of, and as I've alluded to, drug-ligand binding interactions. CD is a very quick, rapid method as long as you have a chiral element to it. Just to continue the theme of chirality, if you were to look at L-alanine versus D-alanine, in CD spectroscopy, be aware that people prefer to refer to these. They're exactly the same as L and D, but they're referred to as R and S. I've forgotten what R stands for, but S stands for sinister. Always remember that one. These are just meaning the same thing, that these are two isomers of each other. And if you ran a CD spectrum of these two isomers, you see mirror images within the CD spectrum. So one will absorb the left-hand light and give you the black spectrum, and the other one will do exactly the reverse and give you the mirror image shown in blue for the right-handed circularly polarized light. So being able to differentiate isomers is a really important thing, including in biopharmaceutics, because as you know, isomers of different drugs. Isomers, oh God. Drugs can be isomer specific, and one isomer will work and the other one won't. So CD is an excellent way to pick out the right isomer in a mix, a racemate mix. Okay, so I'll return to that theme in a moment, but so a quick summary of the advantages and disadvantages of CD spectroscopy is shown here. So if you have a macromolecule, you expose it to some treatment, could be heat, could be a chemical. You can probe changes in the macromolecule itself by looking at it with CD. Or you can look at its interactions with small molecules, so a drug binding to a protein, for example. So especially achiral ones, so I've put that in because as long as if you're looking at interactions, as long as one of the molecules is chiral, then if there's an interaction, you're likely to see it. Because if your protein is chiral and your drug is not, and they interact, there may be a conformational change in the protein resulting from drug binding, and that is what you detect in the CD, the conformational change. Far UV will detect changes in the secondary structure of a protein. CD in the near UV region will tell you what's happening to the aromatic residues as a result of that conformational change, i.e. it reflects tertiary changes, conformational changes upon drug binding. So it doesn't matter if the drug is achiral necessarily, it doesn't eliminate using CD. So the advantages are that it's very quick to perform. We've already mentioned that it's uniquely sensitive to the asymmetry of the system and for developing enantiomerically pure drugs. So if you have a racemate mixture of two drugs and you want to select for the one particular isomer, you can monitor the success of your racemate separation into its two isomers using CD very well. You're dealing with molecules in solution phase, which is always advantageous in biopharmaceutics. It requires far lower concentrations than you need for NMR and crystallization. What sort of concentrations do you think you need for crystallizing a, let's say, a protein molecule? Any advance on 30 mg per mil?
10 mg per mil?
Yeah. 30 would be just completely ideal, but not always practical because it's not always possible. If you can get it to 30 mg per mil, wonderful. 10 mg per mil is also right and would be excellent and ideal for that. Often we can't get proteins at that sort of concentration for crystallization. So sometimes we just can't get that structural information, and CD will give us limited structural information with much lower concentrations. So you can use concentrations in far UV of roughly, 6 micromolar, and in far UV 20 micromolar should give you some meaningful data. So you require much lower concentrations and you get limited structural information back. CD can be used for any size of molecule, but it won't really tell you much if you've got dimers, trimers, oligomers, that sort of thing. It's not going to report on unless there's a conformational change upon dimerization or something, often. Though we'll see there are exceptions with the carbohydrates in a moment. And ligand binding is to molecules. CD is quite good at telling you more than some other biophysical techniques. So isothermal titration calorimetry, ITC, will give you a KD value for the binding strength interactions between two molecules through the absorption of heat or the evolution of heat from the reaction of interacting. That tells you something about the strength of the interactions. So you can get all that from CD as well, but in addition, you can monitor different groups of different groups on your molecule, your different aromatic residues and so on. So you could argue that you get more information back from CD than you might from some other techniques like ITC. Depends what you're after. It always is, to choose which technique. So disadvantages, you can get some limited structural information, but of course it's nowhere near the amount of structural information you're going to get with NMR or X-ray crystallography for solving structures to atomic level. You can't do that. So it is used widely in biopharmaceutics for looking at drug interactions in particular, as I've already mentioned. So this is just to reiterate what I've already said, I think, really, about separating racemate mixtures of isomers or enantiomers. So ibuprofen would be a classic example of that. It happens to be a chiral molecule. We know what ibuprofen is used for. It comes in two forms when it's manufactured, as the R and the S form. This is the D, this is the L. And the one that is biologically desired and active is the S form. That's the one that's desired, and this one is not. So these two have to be separated. And CD can be used to monitor that separation process. How do we separate, by the way, racemate mixtures? If you got a mixture of isomers and you wanted to separate it for drug, to pull out the drug you want and not the useless one that's just going to compete with the binding site, how would you do that? So CD can be used to monitor it, but the actual process of separating them, anyone know how you would do that?
With another chiral molecule?
Yeah, possibly. How would you develop that? How would you separate the two isomers? You're right. You're right. Do you know how you would do it in detail?
Maybe get a solution of the racemate with the other chiral molecule, and then like you could get different like polarized light and see the complexes they get.
Yes.
And see the possible cases.
Yeah, you can monitor it, which is what this slide, is that what you mean? You can, yes, fire a CD at it, and you can tell by how much left-handed light is absorbed compared to how much what right-handed is absorbed. Yes, and as you separate them, one is going to become predominant over the other. So you can, this is exactly the point, is that CD can be used to monitor your racemate separating process. But how do you do the process?
Can you get the stationary phase which interact with one of the molecules?
Yes, yeah, great, that's great. So chromatography, but you have to prime up one of your isomers in some sort of chemical reaction to derivatize it so that it can be attached to a stationary support for chromatography. Yeah, is that what you meant? That's what I took, yeah, spot on.
Could you use enzymes?
Yes. Did you say enzymes?
Yeah.
Perfect, yeah. You can use enzymes, and of course we know that some enzymes are very isomer specific. So you can convert one of your isomers, presumably perhaps your unwanted one, with enzymatic conversion, yeah. Great answers. Any others?
There's a chemical thing with crystallization, but I forgot.
Oh, well done. Yes, that's the other one. Okay, so yes, what you do is you start off with a crystal of your D form and a crystal of your L form, and you have a supersaturated solution of your racemate mixture that you want to separate. And all you do is seed that crystal into your mixture solution. And what happens is you get a rapid production of crystals of the appropriate isomer just forming in the solution. Yeah, exactly, spot on. Yeah, okay. So D crystals. So you just seed it and away it goes. And L crystals. So has somebody else explained that, or did you know from before you came here, or
Crystallization.
from crystallization? Oh, you didn't know the crystallization one. Okay, well, you covered it all between you. That's good. That's great. So CD is a great technique to go with that to monitor. Okay. Yeah, I think I've said everything that's on that slide there. We've discussed that. Is that all right? Everyone understands where CD fits into that? Okay, that's good. Right, so let's turn to some specific biological molecules. Protein, for example. What can CD do for protein studies? So we can look in the far UV, which is usually about 160 to 260 nanometers. The far UV will report on changes, structural conformational changes to the secondary structure of the protein. And that's because the chromophore is the peptide bond. And it's the environment of the peptide bond which it's reporting on. Okay, so for example, well, let's start with the first picture here. What it's telling you is that the spectral characteristics are different depending on what the secondary structure of the proteins are. So you can differentiate alpha helix from beta turn proteins, from beta sheet, and these unusual 3-1 helix or polypro, where you have lots and lots of prolines which form a rather unique helical structure because of the way proline in a peptide introduces very significant twists in a protein. So if you've got multiple prolines, then you get a unique spectrum shown here. And then if your protein is degraded and it's now lacking secondary structure, then you see something like the dashed line, which is running along more or less close to the 0 line. Okay, so if it's a shallow spectrum close to the 0 line, you can suspect that your protein is perhaps denatured and it's lost its secondary structure. Whereas if you get nice deep spectral traces in the positive and in the negative regions, you can conclude that your protein has secondary structure. So you may have put your protein through all sorts of harsh biochemical, chromatographical, chromatography-based purifications. Myself, I work with membrane proteins, so I have to shove loads of detergent in there to solubilize them. These are damaging chemicals. And at the end of the purification process, I want to know that my membrane proteins are still intact. They still have secondary structure to do something meaningful with them at the end. And I use CD to monitor whether my protein has come out the other end as something that is likely to be active, or at least that it still contains its secondary structural conformation, which is a clue that it's likely to still be folded and active. So CD is a rapid way to check for structural conformational integrity in terms of the secondary structure. Now, coming back to the peptide bond is the chromophore, which produces the transitions. So 210 and 230 and 190 for the alpha helix is a common theme in alpha helices. These are the transitions. N to pi star and pi to pi star transitions are responsible for giving those spectral characteristics. So all proteins have peptide bonds, so why don't they all appear the same? The reason for that is, of course, literally their secondary structural arrangement. So, for example, alpha helices are shown here on the right, which depend on 3.6 residues per helical turn. That's characteristic, and the characteristic comes from the hydrogen bonding arrangement between the different helices. So the hydrogen bond on the nitrogen of one amino acid, hydrogen bonds with the oxygen, is it? On the amino acid residue, which is 4 residues further up. So the hydrogen bonding is cross-linking and forming that alpha helices, that helix. So it's hydrogen bonding which forms the alpha helix, which disrupts the environment of the peptide bond, which is shown here. Carbon-nitrogen peptide, and it's shown, I tried to highlight it here. Here's the peptide bond here, and you can see with the proximity of the hydrogen bonding that this changes the environment of that CN bond, and that is what shifts these different spectra to look different to each other because their secondary structure is different and hydrogen bonded differently. Is that okay? So that explains why these are different, though they all have peptide bonds. They're all in different environments. So I've mentioned you can check structural integrity of your proteins. You can make mutant proteins, perhaps, and check that they are still stable and structurally intact by CD. You can look for natively disordered regions in proteins because, as mentioned, if they're disordered and denatured, you'll see a shallowing of the spectrum across, and it'll just follow the zero line. You can look at protein folding, so you can start with a spectrum that's along the zero line, and as you initiate a folding process in the protein, you'll see it acquire its secondary structure with time, and you can examine that folding process. And finally, with reference to biopharmaceutics in particular, a theme that I'm driving home here is that ligand binding, drug binding. As long as the drug affects the secondary structure of your protein, you would use far UV studies to observe that change in the secondary structure. So some drugs do that, and they have that's quite a change to change the secondary structure of a protein due to drug binding, but it happens, and it can change the secondary structural conformation quite significantly. So it might still be overall, for example, alpha helical, but it will drop the intensity of the alpha helical nature, because as the conformation shifts, it might unwind one of the alpha helices in the process. So a drop in the overall intensity of the spectra. Is that all right? Okay. So that's proteins. In the near UV, you can also study proteins in a different way. Instead of secondary structure now, you can detect changes in the tertiary structure, the side chains of the aromatic residues, to be specific. So if you imagine the side chains as flags waving around in the environment, if you bind a drug, for example, if that has an impact on the environment of the aromatic side chains, and it shifts the orientation of that side chain flag in the environment, you'll pick it up with near UV CD. So there are different regions of the spectrum that are specific for different aromatic residues. So the phenylalanines are in the 260 nanometer range, tyrosines from 272 to 300 nanometers, and tryptophans a bit later in the higher wavelengths from 280. The tryptophan and the tyrosine spectra overlap, so sometimes it's difficult to tell apart what's happening, but up to 300 nanometers. So specific regions of the spectrum are going to report to you on what the side chains of these different aromatics are doing, plus and minus your drug effects on the protein. So this is reporting tertiary structural changes. So again, you can look at differences between wild type and mutant proteins. You can look at protein interactions and ligand binding once again by looking in the near UV. It just all depends on your protein. So it depends what impact that drug has on the protein. Might be on secondary structure, but it might be on tertiary structure. You don't always pick things up with near UV, because if it doesn't affect the aromatics, you don't see it. The drug could still be binding, it just doesn't perturb the aromatic residues. So there is that to bear in mind. Okay, so how does nucleic acid CD work then? Okay, so these are the CD spectra that you see for 3 different forms of DNA, the A and the B form, and the newly discovered Z form. And in fact, Z DNA was discovered using CD. These are the natural forms you get. So why do you get different spectra depending on whether it's A, B or Z? Because after all, DNA is made up pretty much of the same stuff, regardless. So why do we see the differences? Well, let's first have a look. The DNA, of course, is made up of nucleotides. We've got sugars in there, which are chiral, and we've got bases in there, which are not chiral, and phosphates, which are not chiral. So it's down to the sugars for chirality. And when the bases are very chromophoric, these are aromatics, aren't they, essentially? So they have strong chromophoric groups that will absorb the light, and the sugars have the chirality. So when they're linked together in DNA, you've got chromophoric groups with a chiral molecule, and you can get a composite which is now chiral and absorbing the light. So that's the first thing, and you get strong pi to pi star transitions, and that's what you see around 270 nanometers. So you see that common to them all, that around 270 you see strong spectral characteristics. But in the case of A, B and Z, they are actually different, distinct spectra. So why is that? The reason is that all these spectra that I've just described, those transitions, are completely out-dominated by the helicity, the double helix arrangement in the DNA. So it's a bit like the secondary structure in proteins. So when you've got hydrogen bonding going on to create a double helix, it's the environment of those chromophoric bases and sugars are now changed and twisted, as shown up here, into a different environment, if you like. Their environment is different because of the extra bonding that goes in to make a double helix. So in the case of A DNA, you have a base tilt of 20 degrees and a base roll of 12, and so on. In B, it's different, it's 5 degrees and naught, and in the Z DNA, it's different again. So all those different helical arrangements, they're all double helices, but they are different in terms of the base tilt and the roll, means that you get slightly different spectra. So you are able to distinguish what's going on with regard to A, B and Z DNA. Again, you can do ligand binding studies and look at the effects on all 3 conformations. So it's actually the stacking geometry of the bases which is important for the CD spectrum. That's why they're different. Okay. Right.
Oh, would we then be able to tell the difference between an unwound and a fully compact DNA? Like if it's formed tightly wound with histones and stuff like that, as opposed to just unwound for replication purposes.
Let's think about that. The histone interactions might produce a difference because I'm unsure about whether histones, do they actually interact with the DNA? It's possible if it's very, very compact, but it might not be on a sufficiently compact, a scale of compactness to impact whether it's A, B or Z. So I'm not sure about that. It's good thought though, as to whether it could be different, whether it interacts with the protein and again changes the DNA a bit. Yeah, principle. Okay. Finally, polysaccharides. Can we usefully use CD for polysaccharides? Steve's favourite subject? Well, you can, but you're looking in not just the far UV, certainly not the near UV. Far, we're looking in the vacuum UV region, ultra low wavelengths to be able to see carbohydrates and polysaccharides. Some of the monosaccharide sugars you can see in this region, and indeed polysaccharides, as I've said. Why do transitions occur here? It's because you're looking at lone pair oxygen electrons of OH groups and glycosidic bonds and acetyl bonds in this case. Okay. There are also carboxyl group and acetamido group containing carbohydrates, which you can look more conveniently in the traditional far UV and near UV regions though. So it depends on the polysaccharide as to what groups they've got and whether they can be seen in CD. So it can be done. The vacuum UV is now synonymous with looking at, as I've mentioned, very low wavelengths, and those low wavelengths aren't easily obtained with benchtop CD instruments. You have to go to Harwell, as we'll see in a moment, to get down to those wavelengths, because you need a high photon flux, very strong light beam to be able to work through to get these very low wavelengths. Anyway, let's have a look at what you might see. This is a glucose molecule, glucose shown here, spectrum, shown all the way down to 140 nanometers, which is, it is quite an achievement to get down to that wavelength. So you can see an individual sugar, and if you looked at trehalose, which is glucose molecules bound together as a 1,1 glucosidic bond, then what you see is a very weak spectrum when the two are together dimerized to make trehalose. And the reason for that is the glucosidic bond has a spectral output which is it just happens to be the mirror image of the one that is negative for the glucose. So they counteract each other. So the bond itself gives a spectrum which is way up here at 160 nanometers. It's counterbalanced by the one from glucose. And so what you end up with is a product of the two, which is a very weedy, weak spectral trace, because they've cancelled each other out. I couldn't think of the phrase. They compensate for each other and you see nothing, but there's actually quite a lot going on underneath that. So you can use that to look at breakage of glucosidic bonds, for example, trehalose to glucose transitions perhaps, or the other way around, formation of dimers, of course. And another example, here's our glucose molecule again here. Then if you look at maltose, which is also a dimer, this time with an alpha-1,4 link of glucose monomers, then you see the spectrum change from this to something much more pronounced in the positive CD region for maltose dimers. And then you've got another permutation with amylose. Amylose, as I'm sure you already know, is helical. And it's like loads of maltose units stuck together, and it forms a helical arrangement. And so amylose, whilst it resembles the maltose spectrum, is different again, and the reason for that is the helicity in the molecule, as we've seen for the protein alpha helix and the DNA double helix. If you have helicity, then obviously that changes the environment of the chromophores in these cases and changes the spectrum. Okay. where would you do vacuum CD? As I've already mentioned, getting ahead of myself, you would go perhaps down to the synchrotron, and this was my cue to remind you of your visit to the diamond synchrotron, so sorry about that, but that won't happen. So certainly for that wavelength of range, you would need Harwell to do that, or an alternative sort of facility, such as Steve mentioned down in Grenoble. I'm sure there are plenty of other places as well, Japan and all sorts around the world, but this is our UK version of that. For other applications, there is a benchtop instrument which you can see here, which works for most of the far UV and certainly for the near UV regions. It's very good, and those instruments are becoming very sophisticated now, and membrane proteins, which I work on, are very difficult to produce in any amounts, and therefore I've always gone to Harwell to do my studies of membrane proteins, simply because I can't produce enough of them to use a benchtop instrument in. But benchtop instruments are becoming better now, and I'm starting to consider going over to benchtop. But the synchrotron, as mentioned, it has a higher photon flux, so it can deal with protein concentrations that are much lower than you would otherwise like, and you can get some good signal-to-noise ratios with very low concentrations of protein preps. So that's why I used the synchrotron for that purpose. But as I say, equally, they're much better at getting at these lower wavelengths for your carbohydrates. Okay, that brings me to our 3 examples that I'm going to run through. Can I just check the timing? Are you all right in terms of where you've got to be in half an hour? Hopefully not take as long as half an hour, but this is example 1, where I'm picking up on that example of ibuprofen again, and diazepam antidepressant. So I've just summarised what these 2 drugs are used for, but it's known that both ibuprofen and diazepam could be co-prescribed for individual patients, of course, but it's also known that they both bind to human serum albumin for circulation in the system, and of course that's important for the delivery and the pharmacokinetics of those drugs and how they are going to work in the patient over time. So the question is here, do they compete for their binding sites on the human serum albumin and compete with each other? So they could interfere with each other, pharmacokinetically speaking. So if one outcompetes the other, then obviously the delivery of the other is going to be impeded by the competing drug. In other words, are they compatible for co-prescription or not, is the question, which you can imagine crops up in all sorts of scenarios where new drugs are developed. So CD can answer this as long as I say, you've got one chiral molecule in the bunch. And in this case, we're looking at the competitive binding of achiral diazepam, you don't see a signal, and ibuprofen, which is chiral, as I spoke about earlier on, to human serum albumin, which is chiral. So we've got a mixture here. And the question is, do they compete with each other? So CD has been used to answer that question by, first of all, looking at the characteristic near UV spectrum of human serum albumin, which is shown by the red line here. Okay, that's the spectrum. And then what happens when you add diazepam to human serum albumin? We know they interact, so the spectrum should change. Diazepam is achiral, so you're not going to see any contribution from that molecule, but you will see what happens when diazepam binds to HSA, because HSA will change. So what you're reporting on here is what's happening to HSA, structurally speaking, the aromatics anyway, in the near UV region. When diazepam binds to it, there is a change in the side chains of the aromatics that is sufficient to be able to see a change in the spectrum. And that's what you see with the blue spectral line, is the HSA, the change in the HSA upon diazepam binding. So in this panel, what they demonstrated is that diazepam, as we already knew, binds to human serum albumin. So we've got that one. So ibuprofen is chiral, HSA is chiral. So when you repeat that experiment, here's your HSA spectrum with ibuprofen. We also know ibuprofen binds. And you see that they are indistinguishable spectra, which might, on initial inspection, sound odd and unexplained, but it doesn't mean, just because those spectra are identical, doesn't mean there isn't an interaction. You have to be aware that in the near UV region, what you're looking at is the aromatics. So if the ibuprofen binds at a place that doesn't affect the aromatics, then you're not going to see a change because the aromatics are unchanged even though ibuprofen is bound. So they do see that, which perhaps implies that these two molecules are binding at different places. Well, no, it doesn't, because this does change the HSA spectrum. Okay, diazepam changes it, but ibuprofen doesn't affect the aromatics, but diazepam does. So there could be two reasons for that. It could be that they bind at different places, or they're having a different effect on the molecule in some way, affecting the aromatics just because of their chemistries. So we can't be sure that they're binding to two different places. And then the final experiment to nail whether they do compete with each other is to start off with HSA with diazepam already bound to it, and then you titrate in your ibuprofen. So you have a spectrum. This is the diazepam HSA spectrum here again in blue. And then, once you've got that spectrum, you then add in the ibuprofen and see whether it changes or not. If it doesn't change, ibuprofen is therefore not able to competitively knock off the diazepam off that binding site. If it does change, of course, it means that they are competing with each other, and the ibuprofen is now knocking the diazepam off and changing the spectrum to something else. And that is exactly what we see, the latter. So the spectrum changes from the blue, with the diazepam bound to it, you add in the ibuprofen, and the spectrum becomes just like the ibuprofen spectrum we saw before. So it means the ibuprofen is competing off the diazepam off the HSA. So in principle, this is showing that these drugs are perhaps not compatible to prescribe together because it relies on HSA to circulate them to their site, and they are competing with the same site. Okay. Right, example 2 is development of all carbohydrate cancer vaccines. Why would we want to do that? Well, the answer is that carbohydrates don't elicit much of an immunological response, generally speaking. Have you met that already from some of your lectures with Steve and other people on the module yet? Maybe that's yet to come, sounds, by looking at your faces. Don't know about this. So in general, carbohydrates don't elicit the T-cell response from memory immunity for very long. It's just always been B-cell immunity, which is transient and not great for vaccine development. So what people have tried is linking their carbohydrate antigens, such as the carbohydrates on the that are produced by tumour cells and unique to tumour cells, and they link them to a protein which is antigenic as a hybrid, and then the protein part will elicit a T-cell mediated memory response and hopefully drag that carbohydrate into a T-cell response as well. And that does work, but it's on a very individual basis. Proteins, great antigenicity, generally speaking. So the idea of an all-carbohydrate cancer vaccine is just born out of the difficulty of, if you do link your cancer carbohydrate to a protein which is antigenic and hope that it works in terms of long-term immunity, it's very difficult to accomplish that. It's much more difficult than the literature suggests. So the alternative suggestion is, well, can we elicit a T-cell response by using all carbohydrate cancer vaccines? So this is what this paper is about, whether that can be done, and what the properties are of such a carbohydrate antigen to elicit that long-term response. So one promising avenue is to use in your fusion of your cancer carbohydrate to another carbohydrate, your carrier as it were. That carrier carbohydrate is proposed here to be something called a spitterionic capsular polysaccharide, ZPA, a PSA for short. So a bacterium called Bacteroides fragilis has its own spitterionic carbohydrate that it produces on its surface. It's important for pathogenesis. We don't need to go into that now, what it actually does in vivo, but a PSA is a spitterionic carbohydrate source, if you like, polysaccharide, that can be used to fuse to a carbohydrate from a tumour to elicit a long-term T-cell mediated immunological response. So the amazing discovery was, first of all, that such a polysaccharide could induce a long-term T-cell response. In this paper by Kreisman and Cobb and Kaspar, this was quite a breakthrough, the fact that you could take a polysaccharide and it would induce such a long-term response. And the question is, how is it that PSA can do this and most carbohydrates can't elicit a T-cell response? And CD, as you might imagine, plays a role in answering that question. So just to remind you about T-cell responses and B-cell responses, are you familiar with the difference? That what you have is, if we take our bacterium, Bacteroides fragilis, that's producing PSA on its surface, this unusual spitterionic polysaccharide, taken up by macrophages and presented to MHC2, class 2, where the molecule is broken up into fragments, presented on MHC2 on the surface, and then you have a T-cell receptor which ultimately is responsible for producing T-cells, which is the desirable long-term immunity, and the production of B-cells, which is not so desirable for long-term immunity. So carbohydrates normally only produce this, PSA produces this and this, so we get the best of both worlds, but we get the long-term one, which is what we're really after. So how is it that that can occur for this unique sort of polysaccharide? Well, there are two things about this which are different to your average polysaccharide. T-cell activation depends on two things which are unique. First of all, it's spitterionic in nature, so it has both positive and negative charges on it, which is relatively unusual, belongs to that particular class. And the other thing that was found out about it is that the propensity to make this T-cell response depends on the mass of the PSA fragments. Not too big, not too small. Okay. So what they found is that native PSA doesn't produce much of a response. It produces some response, but when it's in its native state, very large, 130 repeating units, you get some T cell response. If you have fragments which are about 15 kilodaltons, which is about 22 repeating units, you get a great response, much better than the native. So smaller is better, but if you go too small, to 3 kilodaltons, you lose it, and there is no activation of T cell production. So it's a certain mass range which seems to be the key, in addition to the charge that's present on the carbohydrate. So, as summarised, thus the epitope for T cell response derived from PSA is more complex than a single repeating unit. So it's not the charge by itself, because the single repeating unit, in fact 6 repeating units, is going to have loads of charge on it, and it's not inducing the T cell response. So it has to be something to do with the mass, that intermediate size range, which is not to do with the charge. So the proposal is, well, it's a conformational component then, isn't it? So this is what they pursued using CD. So myoglobin's the control, it's an all alpha helical protein. So what they found when they did CD on PSA is that you see a spectrum which is indicative of alpha helices, just like in the proteins. That's completely characteristic of alpha helical proteins, as shown by the myoglobin control. And when they break PSA down into their constituent monosaccharides, then you see absolutely nothing, as you'd expect. So the conclusion, first of all, of course, is that PSA is alpha helical, which is relatively unusual. The other helical polysaccharide that you've heard about so far being amylose, yeah. So this was the first surprise, is PSA is alpha helical. And far UV was used to establish that. And then they wanted to determine, well, why is it the size is so critical? So what they did was an experiment to determine whether that alpha helicity is important for binding to MHC2 and thereby mediating the T cell response. So what they did was they used PSA, which had been chopped up with ozone, and they got fractions which are hardly touched and large, shown by the black region, 0 minutes cleavage with ozone, and then various time intervals at which PSA is exposed to ozone. So at 90 minutes you expect most breakdown of PSA, and shown by the orange components here, broken down to between 0.1 and 1 kilodalton. And they used all these fractionated fractions from the Cephedex size exclusion chromatography to test whether they would bind to MHC2, first of all. If they bind, that's taken as T cell activation. And they also looked at the CD spectrum to see whether it was contingent on the helicity of the polysaccharide. And as you can see, here's the alpha helix in the black spectrum at 0 breakdown of PSA, lovely and alpha helical. And you see some binding to MHC2. Then as we progressively go down the colour banding, showing the smaller and smaller fragments, you can see that by 90 minutes we've got virtually a flat spectrum indicative of total breakdown, or at least loss of helicity completely. There's no helix there in these small bands, and we get no binding to MHC2. But there is a size range in between, optimised at 45 minutes with a purple bar, where you've got some helicity, but you've got maximum binding to MHC2 by that particular fraction. So first of all, they've shown that there is a mass dependency in terms of binding to MHC2, and looking at this, the cutoff is roughly where the orange spectrum is, the 90 minutes. So if it's less than 90 minutes, you've got at least some alpha helicity, and the authors then made the connection that the binding to MHC2 is dependent on alpha helicity of a cutoff of 3 to 30 kilodaltons is the size range where it retains enough helical character to be an optimum range for activating MHC2. Not entirely convinced by it, but they do link up. So, importantly, the orange definitely does show loss of helical character is accompanied by loss of binding to MHC2, so you can argue that one. And therefore, that the helical conformation is important for binding to MHC2 is the conclusion from that data there. So helicity is important. So in creating this all-carbohydrate vaccine, the question is, when you link the tumour carbohydrate to the PSA, will it retain its alpha helicity for MHC2 binding? Okay, so these guys did that. Travic et al. 2014 took the unusual sugar moiety from the TN tumour-associated antigen for this construction and fused it to PSA, a PSA derivative, which is small, to see whether it would retain its alpha helicity. And that's what they did. So here is the sugar by itself, not really doing anything in terms of secondary structural conformation or any other very much. And then here's PSA, the PSA derivative here, again showing its nice alpha helicity here with the black spectrum. And when you fuse the two together, you can see that there is a loss of a little bit, but predominantly the green spectrum is showing that a lot of that helicity is retained. And therefore, they've achieved success in successfully making something which should be great and optimal for activating T cells and getting MHC2 binding for generation of the vaccine. So that's where CD makes its contribution there. The final example is something Steve was alluding to this morning about vancomycin, this small 1500 Dalton glycopeptide antibiotic. So I just want to run through quickly about vancomycin. Time's running against us. So as Steve mentioned this morning, vancomycin is a glycopeptide. It's got two sugar residues on it, a rather unusual one, vancosamine with glucose. And this is linked to some amino acids which are very unusual in nature. It is an antibiotic which is produced in nature by a microorganism. These are unusual aromatics, but great for CD because it is stuffed full of aromatics.
Are you okay for another 15 minutes?
Yes, I should well be done within that.
Okay, because I've missed the recording off the last end of my lecture, so I'm going to record that now.
Should be 10 minutes max.
That should be 10 minutes.
Is that all right?
Okay.
So vancomycin works against gram-positive bacteria because it binds to the peptidoglycan layer of the bacteria. It doesn't have to cross the membrane, it's too big for that. So what it does is it binds to the terminal diala at the end of the sugar chains in the peptidoglycan, little short peptides. So it's a steric hindrance, so vancomycin is a big molecule relative to diala, and what it prevents is cross-linking with other strands of peptidoglycan. So there would be a link between these two, the diala and the lysine of its neighbour, normally in a process called transpeptidation. But transpeptidation is prevented because this large vancomycin molecule is stuck to the diala, preventing that cross-link happening. So the peptidoglycan gets weaker and weaker because it can't cross-link. Just in the same way penicillin works in the same way, except penicillin binds to the transpeptidase enzyme rather than the diala, but it's the same process they're disrupting. So what happens is it gets weaker and weaker, and finally the peptidoglycan can't maintain the shape and the structure of the cell, and it pops. It's just an osmotic lysis of the cells. Same end effect as penicillin, but it just binds to a different place in the process. So that's what the antibiotic normally does. One consequence of that is you get a build-up. The cell is busy producing peptidoglycan monomers to insert into its peptidoglycan layer. So you get an accumulation of peptidoglycan precursors as a result of vancomycin interactions. Keep that in your head because it'll come in again later and be important. So that's how vancomycin works. And vancomycin resistance works, generally speaking, by the bacterium adapting by switching on a resistance mechanism, which results in the production of diala-D-lactate. So it doesn't produce diala-diala anymore, to which vancomycin can bind. It binds diala-D-lactate. And vancomycin cannot bind with the same strength as to diala-diala. It loses a hydrogen bond, and the result of just loss of one hydrogen bond is that binding by vancomycin is 1,000-fold reduced. So the bacterium is home and dry, resistance through putting diala-D-lactate into its cell wall instead of diala-diala, and vancomycin can no longer effectively bind. And the bacterium is free to grow, multiply, etc., free from vancomycin. So that is the resistance. And the question here is, resistance, the way diala-D-lac is produced in the cell means that there has to be an activation of this signal transduction pathway to divert pyruvate into producing lactate, and hence you get diala-D-lactate. This is the mechanism which is responsible for activating resistance to vancomycin. It's called the Van-SR two-component signal transduction system. And the question in this field is, can we be sure what the activating signal for vancomycin resistance is? Is it the antibiotic itself which activates this cascade for production of this diala-D-lactate, or is it the peptidoglycan precursors that I mentioned earlier? So, controversy in the field, and there's evidence for both. So, the research I'm talking about, I suppose, is my own, where to answer that question, we produce the Van-S sensor as a purified protein, membrane protein. Tricky to do, but we managed to produce the purified protein in detergent to solubilize it in solution, and then challenge it with vancomycin and challenge it with peptidoglycan precursors and see whether there's any binding to explain whether it binds to Van-S or not, and hence could be the signal for activating this resistance mechanism. So we did that, and we looked at the purified protein in the far UV region in the presence and absence of vancomycin and found virtually no effect. So if vancomycin binds, it exerts no discernible effect on secondary structural conformation of Van-S. Okay? Then we switched to the near UV region and repeated the experiment. This is the Van-S spectrum by itself, okay, in the near UV region, and you can see already what a dramatic change you get if it's Van-S with vancomycin. You might say, well, vancomycin is very aromatic, this could just reflect the vancomycin here. Does anyone know what the answer to that is and why I would say no, this is a good experiment? Because this is what's known as a different spectrum. So in the coursework, look out for whether people are presenting their data as different spectra. And what that means is they have subtracted the solvent, the vancomycin in my case, in separate spectra. So they run a spectra of vancomycin alone in my case. So all those lovely aromatics that are present in vancomycin, we get the spectrum for that and subtract that spectrum from the Van S with vancomycin spectra. So we get that spectrum, the protein and the vancomycin, and we take away the spectrum for the vancomycin alone. And hence, we take out of the results any contribution by vancomycin, salt, buffer, the solvent for the vancomycin, all the other components that are in there. So look out for different spectra when you're looking and evaluating research papers, that it's a different spectrum. And in our case, this is a different spectrum. So this dramatic drop is not due to vancomycin, it's due to the change in Van S and vancomycin as a result of their interaction together. So they do interact. So we have shown that Van S and vancomycin do bind. So vancomycin could be the signal. So the other thing to eliminate is, well, do the peptidoglycan precursors also do the same thing to Van S, which is the other possibility. So these CD spectra are trying out a variety of peptidoglycan precursor molecules and whether they bind to Van S or not. This is the control, Van S alone, in the near UV, shown here. So we add vancomycin, the red spectrum, and we see the same dip that we saw before in the previous spectrum, as expected. But if you use some of these peptidoglycan precursors, part of the peptidoglycan, they do not change significantly from the control. And it's only when you've got glycopeptide present in these different spectra that you see this drop and this characteristic sign of binding. So in this case, the purple one is teicoplanin. The reason that works is teicoplanin is related to vancomycin. It's an antibiotic homologue, analog of vancomycin that is used in the clinic. So that's a glycopeptide as well, and we'd expect to see that bind if vancomycin does, and it does. And the red one is vancomycin with Van S, and I forget what the grey one. The grey one is vancomycin is present in association with the diala that it binds to. So again, you see the spectral shift. So vancomycin and teicoplanin are involved in binding to Van S. And we can do titrations with Van S to determine what the KD value is, the strength of binding between them, and we find that 70 micromolar, it's given away on the slide about what the strength of a 70 micromolar value for a KD is, and that's very weak because we're running out of time. I won't go beyond that. So you can do titrations, just add in more and more vancomycin. It changes the spectrum increasingly, and you can use that to model and find out what the KD value is that's equivalent to. I won't hesitate over the summary because I've held you long enough. This slide is just to remind me to tell you about the coursework element and to introduce that very briefly. The CD question is based on far UV data, and you're asked to interpret what the data is showing for a number of different papers and different examples of interactions between molecules in the far UV region. And you're asked to provide a number of things, not just interpret the data, but set it in context with regard to what drug we're talking about, what target it's interacting with, and the biology behind it, so that we know that you're getting an understanding of how the CD can be useful for solving different research questions. So it's the background as well, and that's explained in the written version of this coursework, which is on Moodle now, so you can download it to get started. So that's hence this statement is very important. You describe the context and significance of the research to get your marks and have read the research papers that go with it. So you get this set of data, which is looking at a bacterial membrane protein and its interactions with some disinfectants, an anti-apoptosis protein and its interactions or not with an anti-tumour drug called Taxol, BSA as a model for human serum albumin, and the effect of an antipsychotic drug which has more side effects than many antipsychotic drugs, and it's part of an investigation to see why that is. And finally, a membrane protein which is involved in quorum sensing in a bacterium and its interactions with a pheromone. So, that's it. I'll stop there. Sorry to overrun.
Sorry. On the graph, so say you have a protein that has alpha helices and beta sheets. Would you see a difference? Like, would you see two curves, or would it be something that is merged together?
It depends what the proportion of those are. So, a protein can be predominantly alpha helical, and you'll see the spectrum of an alpha helix, even though it's carrying small amounts of other types of secondary structure. It'll take the predominant one and reflect that in the spectrum. If it was 50-50, I think you'd probably end up with something that was very in between the two spectra. Yeah.
For the multi-drug interactions, is CD the most used technique for that?
It's often used because it's so rapid to get a result, I would say. But it's not, of course, the only technique. So if they had any doubts about whether they can see the interaction, because as I've mentioned, you don't always see it if it doesn't affect the aromatics, for example. So you would use, the best rule of thumb is to use more than one biophysical technique to demonstrate an interaction. So you might turn to AUC or ITC that I mentioned as well, as that's the best way to tackle it, is to be 100% sure by using more than one biophysical method. But because it's so rapid, CD to perform, if you've got all your materials and your protein's ready, you can get a far UV spectrum in 2 minutes, and that's replicated 4 times. So 10 minutes, and you've got a very perfect spectrum. Okay, you've got to do all your controls, but you get an answer very quickly. So
you need a synchrotron though, or can you use any CD instrument?
Yes. For getting a quick result, you can use either. Benchtop's just as fast. Yeah.
If you're working for a small medium enterprise and you want to, is it worth buying in a CD instrument, or is it just worth contacting Harwell and applying to get some beam time?
No, benchtop costs about 70k.
Yeah, it's quite a lot for an SME.
Harwell, it's a competitive application process, but so it takes time. So they have to consider your application and then give you the results. So 3 months, maybe 4 months before you get to do your experiment at Harwell.
And businesses can't wait that long, can they?
Depends what your objectives are.